We are starting with genetics of Drosophila development, and to start with Drosophila development we must know the basics about Drosophila body plan. Drosophila body is divided into three portions. The first one is head, then we have thorax, and thorax has paired appendages, and at last we have abdomen. Head is a large segment in itself, and thorax is divided into three segments. First one is T1, which is called prothorax. Prothorax has one pair of legs. T2 is mesothorax, which is having one pair of legs as well as one pair of wings. And third one, T3, is metathorax, and metathorax is having one pair of legs as well as one pair of halteus. Halteus are modified wing structures which will be acting as balancing organs. The abdomen is divided into many segments from A1 to A8. The head region of Drosophila is considered as anterior portion, and tail region or the last segments like A7 and A8 is considered as posterior region of Drosophila body. And we will talk about the development of anterior-posterior axis as well as dorso-ventral axis of Drosophila in a moment. So it all starts with Drosophila ovary. In Drosophila ovary, there are many ovarioles. As you can see, this is Drosophila ovary, which is having many ovarioles, and these ovarioles are having certain cells we call stem cells. Now, stem cells are the cells which divide into two type of cells. One cell is completely identical and which will return into stem cell pool, and second outer cell is slightly differentiated. This is what a stem cell is. So a stem cell will divide into four successive divisions, and these are incomplete mitotic divisions, and we will get 16 interconnected cells. These cells are interconnected by cytoplasmic connections, as you can see. Now, this structure in which 16 interconnected cells are there is what we call as cyst. You can see cyst is this structure right here. Now, out of 16, one cell will be selected as oocyte and remaining 15 cells will be acting as nerve cells. So, how do we select which cell will become the oocyte and which cells will stay as nerve cells? Now, the cell which is having maximum cytosolic connections will become oocyte, like the cell right here. It is having 4 cytosolic connections directly connected with other 4 cells. So, this particular cell will be selected as oocyte through specific signaling, and remaining cells will be acting as nurse cells, as you can see. The oocyte, which is shown in blue color, and nurse cells which are surrounding this oocyte. And in case of the germarium of ovariole of Drosophila ovary, we have germline stem cells from which we got one oocyte and 15 nerve cells. Other type of cells which are present in germarium are somatic stem cells. Now, these somatic stem cells will give rise to certain kind of epithelial cells we call follicle cells. These follicle cells will cover this entire structure, this oocyte as well as nerve cells. So, this oocyte and 15 nerve cells are derived from germline stem cells, and the cells surrounding these oocyte as well as 15 nerve cells, which are these cells, epithelial cells, these are derived from these somatic cells. Now we will start with the genetic mechanisms in axis specification in Drosophila body. Let's first understand the structure of oogonium. So the oogonium has outer follicle cells, and these follicle cells are epithelial in nature. They will produce certain substances which are chorion, which is forming the eggshells outside this whole structure, and it also secretes certain proteins, certain molecules which will along with chorion acting in the eggshell and making the beta-line membrane. So outside or outermost membrane present is beta-line membrane, and inside beta-line membrane we have follicular cells, and these follicular cells will cover these 15 nerve cells and one specific oocyte. Now, what will start to happen is these nurse cells will start producing certain maternal mRNAs, and these nurse cells will start supplying these maternal mRNAs to this specific oocyte. As you can see, these are maternal mRNAs, and within oocyte, these maternal mRNAs will be translated to specific proteins, and these proteins are from maternal origin, and these proteins are important in axis specifications and other developmental mechanisms. Now let's start with anterior-posterior axis. How is this anterior and posterior axis be determined? Fertilization has not taken place yet, and we have a nucleus here which is shifted slightly towards periphery, and these peripheral follicle cells will start expressing one protein which is represented here by this orange color. This protein is what we call torpedo, and the oocyte will start making certain mRNAs which are Gurkhan mRNAs, and these Gurkhan mRNAs will be translated into Gurkhan proteins which are presented or which are shown here by this green color. Now this Gurkhan protein will act as ligand for the torpedo receptor. So this Gurkhan protein will sit on the torpedo receptor and initiate certain kind of signaling, and when this interaction between Gurkhan and torpedo take place, these terminal follicle cells or these terminal follicle epithelial cells will start giving posterior cell fate. So this side becomes posterior side of developing embryo in future. So this Gurkhan torpedo association will make these cells posterior follicle cells, and when this happens, there is initiation of other type of signaling as well. Torpedo Gurkhan interaction will initiate these terminal follicle cells in secreting a certain protein which is protein kinase A signal, and this protein kinase A signal will be transferred to this developing oocyte. So what has happened is there is secretion of Gurkhan proteins by this oocyte itself. The terminal follicle cells has made torpedo receptors. Gurkhan torpedo interaction takes place. The posterior cell fate arises in these terminal follicle cells, and as soon as the posterior cell fate arises, these terminal follicle cells will start making protein kinase A. This protein kinase A will eventually reach this oocyte, and this protein kinase A will initiate or will localize a specific protein which is PAR1, the partition defective. As you can see, this protein kinase A has recruited a specific protein which is PAR1 or partition defective 1, and this PAR1 will reorganize the microtubule cytoskeleton all along this developing oocyte, as you can see in this diagram. So this is the reorganized microtubule cytoskeleton by the action of PAR1, and the negative side or the negative end is toward anterior portion, and the positive end of these microtubules is toward the posterior portion of this developing oocyte. There are two type of motor proteins which are important in this development. The first one is kinesin and second one is dynein. The dynein is negatively oriented motor protein. It will carry cargo from the positive to negative end, means it will carry cargo from the posterior to anterior portion, whereas kinesin will work oppositely. It will carry cargo from the negative to positive, or we can say anterior to posterior end. There are two specific proteins which are swallow and exoparencia. These specific proteins will make complex with bicoid mRNAs, and they will be attached with the dynein, and dynein carries these bicoid mRNAs as well as swallow and exoparencia proteins toward the anterior end of the developing oocyte. Now similarly, the kinesin, which is positively acting motor protein, will carry Oscar mRNA, and when Oscar mRNA reaches the posterior side, it will renucleate the germplasm, which means that it will localize certain specific proteins like Vasa protein, Tudor protein, and certain mRNAs like nanos mRNA. So what is the significance of these two proteins, the swallow exoparencia and Oscar Tudor? The swallow and exoporantia is localizing the bicoid mRNA towards the anterior side of the embryo, and this Oscar and Tudor is localizing the nanos toward the posterior side of the embryo. And these bicoid and nanos will be acting as the major agents in establishing the anterior-posterior polarity in case of the developing embryo. The bicoid is being carried towards the negative end or anterior portion, and Oscar is being carried toward the positive and posterior portion. And Oscar and Tudor is localizing the nanos to positive and posterior portion, and swallow exoporantia is localizing the bicoid to negative end or anterior portion. So we have bicoid at anterior and nanos at posterior as a result of all these interactions. As you can see, we have bicoid mRNA in the cytosol in the anterior portion, and we have nanos mRNA in the cytosol in posterior portion. Now this is the structure which we call micropyle. Micropyle is a structure from which the sperm entry will take place and fertilization will take place. Now this structure is so small that it will allow only one sperm to enter, and there is no need to block polyspermy in case of Drosophila development. So after the localization of bicoid mRNA at anterior side and nanos mRNA at posterior side, they will be translated into their respective proteins, and there will be development of certain gradients. As you can see, bicoid is higher at anterior and negligible at posterior end. Nanos on the other hand is higher at the posterior end and negligible at anterior end. The swallow and exoporantia, as you can see, is present at anterior end. The Oscar and Tudor, as you can see, is present at posterior end. There are two other proteins. One is this green one, which is hunchback, and second one is this black one, which is caudal. These hunchback and caudal proteins are distributed all along the Drosophila developing embryo. What happens is the localization of bicoid nanos and the uniform distribution of hunchback as well as caudal mRNA has already taken place. Now there is the event of fertilization which is taking place, and the sperm enters through micropyle. Now there is establishment of gradients, the bicoid gradient as you can see, and nanos gradient, nanos toward posterior and bicoid towards anterior, and there will be cellularization, which means that now the follicle cells which we talked about right here, these follicle cells will be gone, will be degenerated, and there will be the divisions of syncytial divisions be taking place. And syncytial divisions is what we mean that nucleus divides but cytoplasm will not divide. So we will see a multinucleated condition. As you can see right here, there are many nucleus but there is no cytosolic or cytoplasmic distinction as well. This is what we call syncytial cleavage. We already talked about this type of cleavage in the cleavage video. After the 13th cycle, to be specific, we get these cytoplasmic connections, or we have peripheral cells all along the embryo as well. Talking about the concentration gradients, you can see bicoid mRNA is in higher concentration at anterior end, nanos is in higher concentration at posterior end. In the middle there is absence of both bicoid as well as nanos mRNA. Hunchback and caudal are distributed uniformly through all along the embryo. But when the early cleavages take place and these syncytial divisions will be taking place, there is a redistribution of this hunchback mRNA as well as caudal mRNA. The hunchback will shift toward the anterior side and this caudal will shift toward the posterior side. And as you can see, the gradient of hunchback is relatively constant throughout the anterior side, and then it falls rapidly and there is negligible amount of hunchback at posterior side. Whereas in case of caudal, the gradient of caudal will fall at slightly consistent rate and there is negligible amount of caudal at anterior side. Now these gradients will be helping in relocalization of other genes and identifying the axis as well as the segmentation of other genes as well. This bicoid will promote hunchback and bicoid will inhibit caudal, which is seen right here. Bicoid is promoting the hunchback, that is why hunchback is seen in anterior portion, and this bicoid is inhibiting the caudal, that is why we have negligible caudal at anterior portion. Whereas nanos behave in a similar way as well. Nanos is promoting caudal, that is why we see caudal at posterior portion, and this nanos is inhibiting hunchback, that is why we see no hunchback at posterior portion. And how it is done? That bicoid mRNA will be translated into bicoid protein, and this bicoid protein will inhibit the translation of the caudal mRNA into caudal protein. Similarly, nanos mRNA will be translated to nanos protein, and this nanos protein will be inhibiting the translation of hunchback mRNA to hunchback protein. That is why we see at the anterior end we have bicoid and hunchback, and at posterior end we have caudal and nanos. This bicoid concentration also determines the fate of various regions of anterior portion. Which region will become head, which region will become mouth parts, which region will be giving you the thorax. It will depend on the concentration of bicoid. Maximum concentration will be around the very much extremity of this anterior region. As you can see, maximum concentration is in this portion only, the extremity of anterior region. So this extremity of anterior region where there is maximum portion of or maximum concentration of bicoid will give rise to head. It will also give rise to acron, preoral structures, labrum, some dorsal hairs, brain primordium. Now, slightly less concentration of bicoid, we can say high concentration of bicoid will give you mouth parts like mandibles, maxillae, labium, and slightly moderate or slightly less concentration of bicoid will give thorax like prothorax. Slightly less concentration will give us mesothorax, and slightly lesser concentration will give us metathorax. And when there is nil concentration of this bicoid, it will give us abdominal segments from A1 to A8 and tail if present. So at anterior end, the bicoid concentration is determining that it will give us head, brain structures, mouth parts, thorax structures like prothorax, mesothorax, and metathorax. And when there is no or negligible concentration of bicoid, the abdominal segments will start, and at the end segments of abdomen there is no concentration of bicoid. Now we will talk about terminal gene effect or the role of terminal genes. We have many terminal genes like Huckleberry, tailless, Groucho torso, acron, telson. But the main genes we will talk about here is acron and telson, because these acron and telson will give us extremity structures like the head structures and tail structures. There is the uniform distribution of one protein which is Groucho, and the role of Groucho is to repress the terminal genes. It will repress tailless, it will repress Huckleberry, and these tailless and Huckleberry are required for the activation of acron and telson. So we can say that this Groucho will indirectly inhibit acron and telson. So wherever there is Groucho, there cannot be acron, there cannot be telson present in that particular area. If we have Groucho uniformly distributed, there is absence of acron and telson all along. But there is something else happening in the follicle cells as well as oocyte membrane. Oocyte membrane will start expressing certain receptors, we call that torsoreceptors. And these follicle cells or these terminal follicle cells will start expressing a certain kind of mRNA which will translate into torso-like protein or torso-like ligand. And this torso-like protein will be secreted by these follicle cells, and this torso-like protein will act as ligand for the torso-receptor. And when the interaction of torso-like protein and torso-receptor will take place, it will initiate RAS-RAF kinase pathway, and the final effector, which is MAP kinase, will phosphorylate the Groucho. Now, what was the function of Groucho? Groucho was inhibiting these terminal genes. It was inhibiting tailless, Huckleberry, acron, telson. Now this interaction, the interaction of torso-like protein with torso receptor, which is a receptor tyrosine kinase by the way, this interaction will phosphorylate the Groucho and make it inactive. And this is happening at the very ends, the very extremities, the terminal follicle cells. near the anterior end as well as posterior end. At anterior and posterior extremities, there is inhibition of Groucho carried out by this torso. So torso inhibits Groucho, and Groucho was inhibiting the talus and Huckleberry, and talus and Huckleberry are very much required for Akron and Telson. So if we inhibit this Groucho itself, the torso is indirectly promoting the activation of Akron and Telson. So at the very ends or at the very extremities of anterior and posterior end, we have activated Akron and Telson, and we have inhibited Groucho. This activated Akron and Telson will give us anterior and posterior structures respectively. Akron is generally responsible for brain and head parts or mouth parts, and Telson is generally responsible for giving the abdominal segments. So as you can see, Groucho is now present at middle, and there is no Groucho at anterior end or posterior end. But you can see there is Akron and Telson present at posterior as well as anterior end. These Akron and Telson are brought about by this torso, which is inhibiting the Groucho at the anterior as well as posterior end. There is another very important concept regarding the behavior of Akron. Akron will only behave like Akron in the presence of Bicoid, or in higher Bicoid concentration, this Akron will behave like Akron and give rise to anterior structures like head and head parts. But if the Bicoid is absent, but if the Bicoid is very low in concentration, then this Akron will not behave like Akron. This Akron will behave like Telson. So at both the ends, at anterior as well as posterior, we have Akron and Telson both present, but the behavior of Akron is controlled by the concentration of Bicoid. If there is the presence of higher concentration of Bicoid, the Akron will give rise to head patterning genes, which are orthodontical, button head, and EMS, empty spiracles, and these head patterning genes will give rise to head and head parts. But at posterior end, where Bicoid is absent or there is no Bicoid concentration present, we have Akron and Telson, but Akron will behave like Telson. Akron will give posterior determinant genes like NRIPS, giant, and Telson organizing genes, and this Akron will behave like Telson and give rise to abdominal segments. That is the most important concept, that Akron will behave like Akron only in the presence of high Bicoid concentration. If we remove Bicoid from anterior end, the Akron will start behaving like Telson. On the anterior side, we have Bicoid mRNA, which is promoting the Akron, and Akron is giving the head region, and this Bicoid is promoting the anterior structures, which are head and thorax. And in case of posterior, we have the nanos and caudal, which are giving rise to abdomen, and Telson is controlling the abdominal segments and give rising to posterior structures. This is the normal development and wild phenotype. But will happen if the embryo is Bicoid deficient. If the embryo is Bicoid deficient, at anterior end, though Akron is present, it will behave like Telson. There will be tail followed by abdomen followed by tail. So this Bicoid deficient embryo will have two tail phenotype. Let's talk about this experiment where we add Bicoid mRNA to certain embryos. The embryo is Bicoid deficient, and if we add Bicoid mRNA to anterior end, it will lead to normal development. Why is it so? Because at anterior end, we already have Akron and Telson. Addition of Bicoid, this Akron will behave like Akron, and there will be head structure development due to this. And at posterior, we did not add any Bicoid, and Bicoid is anyway not required at posterior end, and posterior end, the Telson will give rise to these abdominal segments. In the second case, where embryo itself was Bicoid deficient, we add Bicoid to middle of this mutant, will give rise to the head structures, and the gradient will follow both the sides, so it will have thorax on both the sides. There is Telson, so it is give rise to abdomen segments. But at anterior end, Akron is present, but there is no Bicoid, and if Bicoid is absent at anterior portion, the default fate will be of Telson, and we will be seeing the head at the middle due to this Bicoid concentration, and two tails at anterior and posterior prospective structures. So we have head in middle, and there are two tails at these two extremities. Now, but if the developing embryo is wild type and we add bicoid to posterior, there is bicoid already present at anterior end. What we did is we added the bicoid externally to the posterior end. So anterior structures will be normal. There will be acron giving rise to head and thorax. There will be abdomen, but this bicoid which is ectopically added to the posterior end will start giving the anterior structures at posterior end, because as we saw, posterior end is also having acron and telson. But in the absence of bicoid, this acron is behaving like telson and giving us abdominal segments. But if we add the bicoid to posterior end, acron will behave like acron and will give the anterior structures like head and thorax. There is two head phenotype we will be getting. What we will get is head on both sides, then we have thorax, then we have abdomen, and if tail is present we have tail. So we have two head phenotype. So in summary, if we add the bicoid on anterior end of bicoid deficient embryo, we get normal development. If we add bicoid ectopically to middle portion of bicoid deficient embryo, we get two tail phenotype, one head two tail phenotype. And if we add the bicoid ectopically to posterior portion of wild type embryo, we get two head phenotype. And if we make the embryo bicoid deficient or bicoid mutant, we get two tail phenotype and head formation will not take place. So in this table here we have certain genes like bicoid, exuperantia, swallow, nanos, tudor, oscar, vasa, pumilo. We talked about some of these genes in previous slides, and their mutant phenotype, if we mutate these genes, the mutant phenotype is mentioned right here, and based on these two columns, the proposed function is mentioned right here. You can go on with this table on your own. Now we will talk about dorso-ventral axis determination in case of Drosophila. Already determined the anterior-posterior axis. These terminal cells are posterior, these terminal cells are anterior. And how do we know that which cells will become the dorsal and which cells will become the ventral side of Drosophila body? Initially what happens is all these follicle cells, whether the upper cells or lower cells, all these cells will make a specific protein we call pipe protein, and this pipe protein is responsible for ventral cell fate. So default fate of all these follicle cells, whether these upper follicle cells or lower follicle cells, the default fate is ventral cell fate. Cytoskeleton has already been relocalized, we saw in this diagram here, and due to this organization or reorganization of microtubule cytoskeleton, we saw certain movements which were required in anterior-posterior development. Now similar kind of movement of nucleus will take place, and nucleus will start moving toward anterior side, and as this nucleus start moving toward anterior side, it will reach the upper extremity of portion like here. As you can see, the nucleus has reached toward anterior side, and this nucleus will start secreting or start making certain mRNAs, and these mRNAs are the same Gurkhan mRNAs which will translate into Gurkhan proteins. Now these follicle cells will express again the specific receptors which are torpedo receptors, and Gurkhan torpedo association will take place. As you can see, the nucleus is making Gurkhan mRNAs, and these mRNAs are translating into Gurkhan proteins, and these follicle cells are having specific torpedo receptors for Gurkhan, and the interaction of Gurkhan proteins with torpedo receptors will initiate a certain kind of signaling. There will be synthesis of a special protein. Come to this diagram. As we said, this is embryo membrane, and nucleus has localized towards these upper follicle cells. This nucleus has transcribed the Gurkhan mRNAs which gave rise to Gurkhan proteins. These Gurkhan proteins will diffuse and interact with this torpedo receptor. There will be initiation of signaling, and these follicle cells will start synthesizing a specific protein which is mirror, and this mirror protein is a specific inhibitor of pipe protein. But we said that the pipe protein was giving the ventral sulfate, and if we inhibit the pipe protein, this side of the developing oocyte will become dorsal, because the pipe was giving the ventral sulfate and we are inhibiting the ventral sulfate by this Gurkhan torpedo interaction. So it will give rise to the dorsal sulfate, as we can see right here. These cells which are getting these Gurkhan mRNA interaction with torpedo will get dorsal sulfate, and on the other side there is no such interaction of these torpedo receptors. So these follicle cells will not be making the mirror inhibitor and pipe synthesis will be normal, and by default the pipe protein will give rise to ventral sulfate. How this pipe protein is responsible for ventral sulfate? As we already talked about, embryogenesis has begun, syncytial divisions has taken place. We are around 13 cycle or higher cycle of cell division. So these follicle cells will be degenerated, and after the follicle cells, what was there? There was eggshell and vitelline membrane. So now egg membrane is directly toward the vitelline membrane, and space between egg membrane and vitelline membrane is what we call perivitelline space. Before the embryogenesis, when this was still oocyte, but what difference was there? There was oocyte membrane. After oocyte there was these follicle cells, and after follicle cells we had these chorion and vitelline membrane, and between the egg membrane and between follicle cell membrane we have perivitelline space. But after the embryogenesis, these cells will be degenerated. A pre-vitelline space will directly be adjacent to vitelline membrane. That is important to know. We have developing embryo, and after successive divisions, around 14th cycle, we have these cells which are divided cells of embryo, and we will be looking at this portion. And in reference to previous diagram, we will be looking at this portion. The only difference is these follicle cells have degenerated, and we are directly aligning this portion with this membrane. There is the synthesis of pipe protein. When the pipe protein is synthesized, this pipe protein will result in sulfation of the vitelline membrane proteins. This pipe protein will sulfate these vitelline membrane proteins, and sulfated vitelline membrane proteins will act as docking site for another specific protein GD, which is gastrulation defective. This gastrulation defective is a type of serine protease. This serine protease will cleave another protein which is snake. Now this GD is cleaving snake protein, and snake protein itself is also serine protease which is cleaving another protein which is ester. Pipe is sulfating vitelline membrane proteins which is acting as docking site for gastrulation defective, which is cleaving the snake protein, and the snake protein is acting on ester protein, and the cleaved ester protein, which is the activated form of ester, is also serine protease, and it is acting on a specific protein which is spazole. This spazole protein, when cleaved, will be acting as ligand for other specific receptors. Now there are specific toll receptors present on the egg membrane, or here embryo we can say, because embryogenesis has taken place. So there are specific toll receptors which are present on embryo membrane, and this spazole will act as ligand for the toll receptor, and when their interaction takes place, there is action on Pele protein. Initially the Pele was inhibited by the tube protein, and when the toll signaling takes place, the Pele protein will be released free. Now this Pele itself is serine threonine kinase, so it will function by phosphorylating another protein complex. In the cytosol we have a protein complex which is dorsal, and this dorsal is inhibited by cactus. So there is dorsal cactus complex, and as long as these two proteins are together, the dorsal protein cannot act on its own. It will be inhibited. So this Pele is serine threonine kinase. It will phosphorylate the cactus protein, and it will phosphorylate the dorsal protein. When cactus is phosphorylated, it will be released from the dorsal protein and it will move away. And when dorsal protein will be activated, it can move to nucleus and act as a transcription factor. So the basic idea or the basic nature of dorsal protein is that it is a cytosolic transcription factor. It is inhibited by cactus, and cactus is inhibited by phosphorylation, and this phosphorylation will be carried out by this Pele protein, and this Pele protein itself is initiated by this toll spazole signaling. And when dorsal protein in its activated form goes to nucleus, it will act as transcription factor for the genes which will be initializing the ventral sulfate. That is how this pipe protein will initiate the ventral sulfate. In a summary, the pipe protein is a sulfation protein. It will sulfate vitelline membrane proteins, which will initiate or which will act as docking site for gastrulation defective. Gastrulation defective acts on snake. Snake acts on ester. Ester acts on spazole. Spazole acts as toll receptor ligand. This toll receptor ligand signaling initiates Pele protein. Pele inhibits the cactus by phosphorylating it, releasing the dorsal. Dorsal will go to nucleus, initiate the transcription of genes required for ventralized sulfate. That is how dorsal and ventral axis determination takes place in Drosophila embryo. In summary, we can say that the mirror protein act as inhibitor of pipe and brings about the dorsal sulfate, whereas the dorsal protein brings about ventral sulfate. Pipe helps this dorsal protein bringing ventral sulfate we saw in previous diagram. This is the summary of polarization axis in Drosophila oocyte. We already talked about this anterior-posterior axis determination as well as the dorso-ventral axis determination along with mutant sulfates. Now we will move on to another important topic,